Other people have talked about mousetraps. In particular, there's a man named Ken Miller. He's a professor of biology at Brown University.
Ken Miller is one of your antagonists. He is one that's written in scientific journals about TTSS, yes?
Yes.
And explain what that is very quickly.
TTSS is the type 3 secretory system. It's a subpart of the flagellum. The flagellum, I told you, has to build itself. This thing called the type 3 secretory system can pump out the parts of the flagellum out through a hollow tube where they can assemble themselves. So it can act as a protein pump, or a pump for the parts of the flagellum.
And what he's really getting at is that what he's saying is if we can show that this TTSS is a secretory, what was it, I'm sorry?
A pump.
It's a pump. And it's a building block, okay, of a part of the complex structure that makes up the bacteria flagellum. And his point is, if we see this in other cells or other mechanics within cells, and it's pushing itself forward because TTSS appears in other type cells, then there's evidence that maybe it could have evolved to this place.
Yes.
Now, I want you to respond to that, because I know that sometimes a lot of the folks that are your antagonists get to write and put their opinions in journals, and when you try and respond, your articles are...
They're deemed uninteresting.
Yes, or rejected.
Yeah, they're
which gets to a question I want to ask you. The disclaimer you gave at the beginning, was that your idea, or do powers that be suggest strongly that you give it?
Well, a little bit of both. Powers that be thought it would be a good idea, and I agreed with them, because you have to realize that in the scientific community, I don't get as many laughs as I get here. And my colleagues were getting static because of the crazy guy down the hall from them. And they were getting emails from other people asking if Lehigh University was turning itself into a Bible college, and graduate students from Lehigh who were going out for postdoctoral interviews were being laughed at because I am at Lehigh.
You were on the faculty.
I'm on the faculty there. So
and this is a lot of what the movie Expelled really is about. It's about some of the pressures within the scientific community to, I think what they say is the science of intelligent design is neither intelligent nor science.
Yeah, they do that. They say very strongly, but they don't have any more reason for it than I've been showing you with some of the responses to my
exactly.
To my argument.
So it gets to an ad hominem, or to the man.
Yes.
They make these statements. They call it science in a cheap tuxedo, and yet when the evidence for that claim are brought forward, they don't stand well on the stage up against it.
No, that's correct. I mean, I'm trying to show the best responses to my arguments, and as you can see, they're just not that impressive. But nonetheless, somebody will bang a gavel, as the judge says, and said, "No, you're clearly not scientific. You go away." It's more or less like people in the Soviet Union a long time ago who objected to communism. They were put in mental institutions because if you didn't think communism was the right system, clearly you were not functioning very well. It's very similar to that.
Or you were
science.
Exiled to a very cold, lonely place, which is a little bit of what's happening. I want to come back this a little bit. I want to get Steve back up here to talk about the evidence for intelligent design within the digital code. One last question before we move. How do I get a subscription to that Cell magazine? It just looked absolutely riveting.
Just write to MIT Press.
Okay, thank you. So that was flooding in on our text line, and I wanted to go ahead and get to that. If I could. All right, let's yield to Steve, and then we'll come back up, and we might look at Mr. Miller's tie clip, or do you want to give a quick explanation of what that slide was, since they got a peek at it, and will Steve come out here and be ready to go as soon as you're done?
Yeah, Kenneth Miller says, well, I can take away a piece, and it won't function as a mousetrap anymore, but I can use it as a tie clip. Yeah, you can, but you can't use it as a mousetrap anymore, and that's the point of irreducible complexity. And I was going to go into great detail about that, but I guess I won't.
All right. Well, we're honored that you're here. One of the One of the reasons I'm just grateful that Mike is here, Mike is the father of 9 children, and so, clearly he's figured out the lack of complexity in the mating process. And that's a whole lot more fun than playing with mousetraps. Amen, brother?
She said this is a church time.
Let's, with that, Steve, talk about the evidence for design inside the code. And why don't you do this? Build this bridge, because when most of us laymen think of a black box, we think of that which is indestructible inside a plane. And that's not the black box that Mike was talking about. He was talking about Darwin could not see what was inside the cell. He believed that, in fact, if the cell was as complex as it was today, he may never have written the book. He would have surrendered at that point and saying, there is such genius inside of that, it, in fact, violates what he wrote in that one quote that you had from Origin of Species. So let's go further now, not just in the complex machinery that Mike talked about. Let's talk about even a step beyond, which is the digital code embedded in that. There's a segue.
That's necessary to build it.
There we go.
Absolutely. And hey, Mike, we're not used to getting that kind of a reaction. How did you warm them up like that? All right, I'm here to talk about the digital code. I hope we get to talk a little bit more about that type 3 secretory thing later, because I've been working with one of Mike's colleagues who's a microbiologist who's been defending Mike's honor, because there's a genetic story behind that objection that I think has shown Mike to be entirely correct, but maybe we can come back to that in Q&A. My job tonight is to talk about I think one of the most fascinating discoveries in the history of humankind. Not just science, but it is the discovery that Watson and Crick made of the digital code in DNA. The first thing they discovered in 1953 was the beautiful double helix structure of DNA. We all know this structure because it's a cultural icon now. We know that DNA, you convict criminals with their DNA. We see DNA on all kinds of things. The double helix is an icon, but it's a beautiful structure. But more important than the structure that Watson and Crick discovered is the mystery that they solved and the mystery that they created. First line of the book that I've just completed is this. It's when Watson and Crick discovered elucidated the structure of the DNA molecule, they solved one mystery, but they created another. The mystery that they solved was the mystery of where hereditary information arises. Since ancient times, people have known that like begets like, that children are like their parents, that organisms are able to copy and replicate themselves in very similar form. And scientists have known that there must be some kind of signal, some sort of memory that allows organisms to reproduce themselves. But where does that memory, that information, that signal reside? When Watson and Crick discovered the structure of DNA, there was a hint in the structure that suggested that the DNA not only has this beautiful helix, but it is embedding some kind of information down its spine. It was suggested by Watson and Crick in their very first paper that this might be the case. But in 1957, 4 years later, Francis Crick, having taken stock of all the developments that were going on, not only in the DNA chemistry world, but also in the world of protein chemistry, put forward what I think is really the basis of modern biology, which is the sequence hypothesis. And in it, he said that there are 4 chemical characters along the spine of the DNA, they're called bases or nucleotide bases, that function just like alphabetic letters in a written text or digital characters in a piece of machine code or software. This is known as the sequence hypothesis. According to Crick, it's not the chemical properties of these bases that is important. Rather, it's their arrangement. Just as it's not important what the weight or the composition of these Scrabble letters is. What's important is the way they're arranged to spell out a message. It's the sequence of the arrangement that it determines the function, not whether the letters are put on wood or tile or plastic. This was the sequence hypothesis. And here's a picture of the DNA molecule. You see the double helix shape. That's made of the sugar phosphate backbone on the outside. That's the medium of the genetic text, the genetic information. But the information is stored on the inside of the molecule in virtue of the specific arrangement of these particular characters. We represent them with A's, C's, G's, and T's. There are 4 different chemicals, and it's the arrangement of those chemicals, again, that determines the function of the molecule. Now, that's all well and good, but what exactly does DNA do? And Watson Crick said that the characters function like alphabetic letters in a written text or like digital characters in a machine code. That's great, but what does the machine code do? What do those instructions tell the cell to do? Well, right as Watson and Crick were elucidating the structure and the function of DNA, well, the structure of DNA, there was a parallel development in molecular biology in the area of protein chemistry. It started in about the same time, 1952. A scientist named Fred Sanger was discovering about the protein insulin. Other scientists began to discover the 3-dimensional structure of proteins, and that they discovered that they were immensely complex 3-dimensional shapes. It defied everybody's expectation. They thought proteins were going to be simple things like hat boxes stacked up on top of each other. The ultimate secret of life, after all, should be law-like and orderly. But proteins weren't like that. We know that proteins do, and they knew that proteins did lots of important jobs in the cell. You can think of proteins as like the toolbox of the cell. Inside the cell, or in a toolbox, you've got a hammer, a wrench, a saw, you've got a plane. All these things perform functions, and they perform the specific functions they do in part because of the specific shapes they have. The same thing is true of proteins. They have very irregular, complicated shapes, and in virtue of those shapes, the proteins are able to do specific jobs in the cell. Here's just a simple illustration. You have a protein that acts as an enzyme. It catalyzes a reaction. In this case, a reaction that breaks apart 2 sugar molecules. But the ability to break apart that disaccharide depends on a very precise 3-dimensional fit between these 2 molecules. The disaccharide nestles into those 2 grooves in this particular enzyme. And everything in biology works according to that same kind of specificity of fit. Proteins make up the structural parts of those nanomachines that Mike Behe was talking about. All those parts in the bacterial flagellar motor and other nanomachines are made of proteins. They fit together and form the machine because of very precise hand-in-glove fit between the parts. Same thing is true for enzymes catalyzing reactions. Same thing is true for proteins that help process the information in the cell. All proteins function because of this very precise 3-dimensional specificity. How do they acquire that specificity? Well, we now know it has a hand-in-glove fit, another take on specificity. We now know that proteins acquire that specificity because of the way their individual subunits are arranged. Now, the subunits of proteins, that sounds like a big scientific word, but hey, I've got snap-lock beads tonight. Ages 2 to 4, it says on the box. Okay. All my best visual aids are stolen from small children, you know. Okay. Okay, here's a protein is a long chain-like molecule that folds into a very specific shape. And it might fold differently depending on the arrangement of the subunits, the amino acids. So each of these colored beads with a different shape represents a different amino acid. And you can see that there's a lot of different ways that we could arrange even just these few beads. Depending upon the arrangement, the shape will be different. It will fold in the protein, or the chain will fold into either a functional protein, or it may lie limp and do nothing at all if the arrangement isn't correct. So the amino acids, and there'll be a test on these equations afterwards, the amino acids are sequenced in very particular ways so that the protein folds in the right way. Get the sequencing wrong, you don't get a functional protein. So Francis Crick, in fact, said that he likened the amino acids to those old typeface blocks that are used in the old newspaper headlines. They all have the same basic chemical structure, but there's a different sticky-outie with each one, a different font. And the arrangement of those sticky-outies determines the, more technical terms, sorry, and it determines the folding and therefore the function. Okay? So we like to say that amino acids have a property, or proteins have a property called sequence specificity. If something has sequence specificity, the function of the whole is determined by the specific arrangement of the parts. We find that in language, we find that in computer codes, we find it in proteins. But the question is, how do proteins acquire that specificity? And this is where Francis Crick's sequence hypothesis came in. According to Crick, the characters along the DNA spine function like alphabetic letters, but what they're spelling, what they're causing to happen is the sequencing of amino acids, which in turn cause the proteins to fold and therefore produce a functional tool in the cell's toolkit. So information on DNA is transcribed, translated, and produces the functional outcomes, the protein machines that the cell needs to survive. So you've got a form of functional information, sometimes referred to as machine code or assembly instructions in the DNA molecule. Now, this is what I call the DNA enigma. And I said at the beginning of my book that Watson and Crick solved one part of that enigma. They solved the question of where biological information resides. It resides along the spine of the DNA molecule. They also helped us with the sequence hypothesis to understand what that information does. It directs the building of proteins, but their discovery raised another even more profound mystery. And that mystery is, where did that information come from? Where did it come from? Now, that's an important mystery for two reasons. First, because we know in our own digital world something about information and its importance. I actually used to ask my students, if you want to give your computer a new function, what do you have to give it? Software, code. Well, the same thing is true in life. If you want to build a new organism from an earlier, simpler organism, or if you want to build life in the first place from simpler chemicals, you have to arrange the parts in very particular ways to make functional organisms. To provide new biological function, you also need information. Same principle is in the software world. And that is a profound code is necessary to build new form and function in biology, just as it is in high technology. Now that raises creates a profound dilemma, because there's a question that scientists almost universally agree has not been solved by any materialistic theory of evolution. We can debate about whether Darwinian evolution is true or not, but I'm going to set that aside for tonight and look at a more fundamental issue. If you think of Darwin's great tree of life as representation of the history of life, all the things out on the branches, the giraffes and the dolphins and the birds and the fishes and us and plants, all of those things are the modern forms of life. But if you go back far enough, according to Darwin, you get to the simple organism that is the progenitor of all life. And he and other scientists have usually conceived of that as some kind of simple cell. Sometimes this is called amoeba to man evolution. That's not quite accurate, but you get the idea, okay? But the question that is raised by the recognition of the centrality of information is one, how do you make those transitions from one form to another, given that you need a lot of information, but to provide any new function, any new form? But an even more fundamental question, and which is the one I'll talk about now, is how do you get life in the first place? Life depends on information. DNA runs the show in even the simplest cell. It directs the construction of the proteins that even the simplest cells need to survive. So where did that information come from? Watson and Crick did not answer that question, but Crick in particular appreciated the gravity of the question. Now, I first came across the second question, what I call the DNA enigma, here in Dallas. I happened to be working here in the early 80s. I was right out of school. I was a geophysicist for an oil company. I learned to speak some good oil company Texan. I was looking for all out in the Gulf. Okay. Yeah. And you know how we spell all down here. It's A-H-L, all right? And so I had no inkling of this debate. I wasn't involved in this kind of thing. I was doing practical, applied science. I was looking at doing digital signal processing of seismic surveys. Interesting stuff. But one day in February of 19... That's the other anniversary. February of 1985, I walked into a conference literally off the street. I'd heard about the night before at the Dallas Hilton downtown. It was a conference that had been funded by a Dallas businessman named William Garrison. If he's here tonight or in town, all I can say is you have no idea what you set in motion. And Mr. Garrison funded this conference. It was a conference of people who held a materialistic worldview and people who held a theistic worldview, but these weren't ordinary people. These were Nobel Prize winners. They were the top cosmologists, origin of life scientists, and people who did neuroscience. The three questions they were addressing were the origin of the universe, the origin of life, and the origin of human consciousness. And the conference literally changed my life. I walked in, I sat down, and there was Alan Sandage in the first session announcing how he had become a religious believer because of the evidence of the new cosmology which pointed to the beginning of the universe. He'd originally been slated to speak on the side of the materialist atheist position and had unceremoniously walked across to sit with the other guys. In the next session that afternoon, there was a discussion of the origin of life. There was some apparently young upstarts in the discussion, a local chemist named Charles Thaxton, who had written an important book called The Mystery of Life's Origin. And he was on the panel with Walter Bradley, a very established professor of thermodynamics and polymer scientist from Texas A&M. On the other side of the panel were leading origin of life researchers. But someone had joined Thaxton and Bradley that was completely unexpected as well. His name was Dean Kenyon. He had published the leading chemical evolutionary theory about the origin of life in a book called Biochemical Predestination. It was the leading college-level text on how life started from simple chemicals. At this conference, he announced publicly for the first time that he no longer believed his own theory. In fact, he explained why it wouldn't work, and he went on to express support for something he called the design hypothesis. And he said that he agreed with Thaxton and Bradley that the information that was required to build the cell pointed powerfully to a designing intelligence. Now, when he said that, well, you can't say this in a church, but something broke loose. I mean, it was unbelievable. The reaction to this was part of what intrigued me, because up to that point, everyone agreed, there was no doubt about this, that the state of origin of life biology was in a state of impasse. Everyone agreed that there was no undirected evolutionary process that could explain the origin of the complexity of the cell, and in particular, this crucial information problem. By the way, nothing has changed on that. If you saw the movie Expelled last year, you saw at the very end of the film, Richard Dawkins, who is not known for his restraint and expression of support for evolutionary theory, acknowledged that no one knows how life first originated, and what he meant, no one knows from an evolutionary point of view. I was a young scientist. I heard this conversation, and I was absolutely intrigued. And afterwards, a local guy introduced me to Charlie Thaxton. And Thaxton was kind of unassuming and friendly. I thought, man, he'd been to Harvard. I was really kind of intimidated. But he invited me to come see him after work. And we started talking, and over the course of the next 6 months, he mentored me. And I learned more and more about this subject, and I couldn't wait to get to grad school to study it. And fortunately, the Atlantic Richfield company made a way for that to happen by laying me off. It was, if you remember 1985, if you're oil company people, you remember what happened to the price of oil, and all the young bucks just were given very generous but severance packages. So a year later, I found myself on my way to England. I was fortunate enough to get a Rotary scholarship from the good people here in Dallas, and I was in Cambridge my first year, and that's where I stayed to end up doing my doctoral work, and I worked on this question of the origin of life. I was intrigued by Thaxton's ideas, but he floated them not in a full book-length treatment or an argument. He floated them in a short epilogue, making a suggestive, kind of intuitive case that information seems to be the kind of thing that comes from intelligent agents. And he said, we know that based on our uniform experience. But he didn't develop this, and I was fascinated by this. And I was also fascinated by this general question, this DNA enigma. Here we had, in the shape of the DNA, the information in life, we had a feature that everyone agreed looked designed. Digital code, even Richard Dawkins says it's like a machine code, and it's uncannily computer-like, he said. But no one had an undirected process that they even claimed could mimic the powers of a designing intelligence to produce that appearance of design. So if you've got an appearance of design and no designer substitute mechanism to explain it, maybe there's evidence of actual design. And I got intrigued. But I wanted to know about the other theories. Were there other approaches that could explain this? And as I began to work on this topic during my PhD research in England, I started to realize, what I came to realize was that this DNA was deeper and far more profound than I had any inkling of when I first encountered this conference or even in my discussions with Dr. Thaxton. What I discovered is that there were three basic approaches that scientists had taken to try to explain this DNA enigma. And the first was just the kind of the raw bootstrap, seat of the pants. Let's try it by chance. And there was a famous French scientist who was a colleague of Francis Crick's, Jacques Monod, and he wrote a book called Chance and Necessity. And he said the basic job of the scientist is to explain everything by reference to chance, random variations, random processes, or necessity, by which he meant natural laws. If I drop a wallet here and it falls to the ground, we'll say that happened because of the law of gravity, and scientists will say that, therefore, that happened by necessity. It was lawful. It had to happen that way. And Monod basically said that's what scientists should do. We should explain things by chance and necessity. And so I thought, well, how have scientists fared at explaining the DNA enigma, the information necessary to produce the first life, by reference to chance, necessity, or as Monod said, sometimes we combine them, the combination of the two. Well, so let's look at these briefly, and you'll see why I think this is such a profound mystery from the standpoint of the materialistic evolutionary approach to the origin of life. The first approach is chance, and there's a basic problem here that was almost universally recognized, and it's the problem of the complexity of the cell, but there's a mathematical dimension to it. If you're a person of mathematical, computational, computer background, you know about combinatorials and well, let me illustrate. If you've got, back to my snap lock beads here, these represent the amino acids. There are 20 possible protein-forming amino acids that could attach to a growing chain that would build a protein at any given site. So if you have just two amino acids linking together, how many possible combinations are there? I heard a couple of math guys say 400, and that's right. Our intuition is always, it's 20 plus 20, right? But no, if you think that each one can combine with 20 others, and there are 20 possibilities at each site, it's actually 20 times 20. Now let's go and let's add another amino acid to the chain. Now how many possibilities are there? Man, this is a smart audience. Okay. I was still starting to rev up the mental math, Okay, so now the fourth, what do we get? 160,000, right? Okay, and on and on. What's happening to these numbers? Are they building are they going up in an additive fashion or a geometric or exponential fashion? Okay, exponential. Now, I'm going to look really smart because I already figured this out. Okay, all right. A modest length protein is going to be in the order of 150 amino acids long. The cell needs many proteins that are much longer than that, the gigantic polymerases, for example. But let's just take a modest length protein of 100, 150 amino acids long. Now, think of the combinatorial problem that we have. That's the number of possible arrangements of amino acids that you could generate. 20 times 20 times 20 times 20 times 20, out to the 150th power. 20 to the 150th is also, now this is where I get to look smart, 10 to the 195th power. I worked that out on my little calculator beforehand. But the point is, that's an unimaginably large number. Now, you can quibble about that, and you can notice that there's more than just one functional protein, and that's correct. So that adjusts the numbers downward. But then you also have to take into account that there's only a certain kind of linkage that can take place chemically, and the odds of that are only 1 and 2 at each site. And there's only one kind of amino acid, a left-handed, not a right-handed kind. And so when you take it all into account, here's the bottom line. The number of combinations of possibilities vastly dwarfs the number of possible events there could have been since the very beginning of the universe, since the Big Bang. There are only 10 to the 80th elementary particles. There are only 10 to the 16th seconds since the beginning of the universe. There's only so fast that interactions can take place between molecules. So think of it this way. All the number of combinations that could possibly be represent a vast haystack grander than the size of the entire universe, and what we're looking for is something like a tiny, tiny needle smaller than the smallest elementary particle. And we have to find that in a blind, undirected search, all right? Most scientists working on the origin of life have realized that the complexity of the cell, the combinatorial complexity, is so great that we will never, ever find life in that way.
Through time plus chance.
Through time plus
What
chance.
you're saying is statistical improbability giving the grandest, number that is presented as a probability for the existence of the universe, which is right around 13.7 billion years right now, I've read recently, correct?
Right.
And so you're saying that there's no way for just this one expression of creation to have taken place statistically within that time period, given the immensity of the probability.
The odds are always better that it didn't happen by chance than that it did, and they're immensely in favor of that it didn't. The probabilities are so small.
And what's happening and so exciting inside the black box, so to speak, is what we're seeing is that scientists, everywhere they go, they keep seeing more and more overwhelming evidence that there is just no way that this could have evolved or happened. The numbers are so grand, it takes not just a tremendous amount of faith, but it goes against all reason, all probability, and all really scientific possibility that it could have happened that way, and yet they still defend it vehemently.
Right, because there are some other possibilities, but let me turn the corner from the negative to the positive, okay?
And I just want to make you aware, I know David's here. It is 8:40.
8:40.
And so we need to get him out here.
5 minutes, and I'll tell you the end of the story.
Okay.
And then we'll get him out.
Go for it.
Okay.
I'll stay here with you.
There's two other... and he's a big guy. I'm not going to mess with that. Okay. Let me turn the... There are other possible purely naturalistic approaches. In my studies, I found them equally inadequate. And if you want to know why, there's a big fat book that we're advertising in the back that's going to be out in June called Signature in the Cell. But I want to tell you the positive side of the story, and this will segue to David, okay? And that is that I had an epiphany because one of the things I want to know is, was Dr. Thaxton right? Could this be made a scientific conclusion, the idea of intelligent design? And when I got to Cambridge, one of the things I began to study was how do scientists reason about events in the remote past? And I found the works of the masters. I found the works of Charles Darwin. And I found the works of his mentor, Charles Lyell. And they had a simple principle, and it was that if you want to explain events in the remote past, you should invoke causes that are known to produce the kind of events that you're trying to explain. Makes sense? You want to explain volcanic ash? Don't invoke an earthquake, invoke a volcanic eruption, right? And so I one day found the front piece on Lyell's book, okay? And this is what it had this amazing subtitle, and it put the light on for me. It said, principles of geology being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation. That's how we got to go about things. We try to explain things by reference to causes that we know can produce the effects in question. And I asked myself a simple question. What is the cause now in operation for the production of digital code? What is the cause now in operation for the origin of information? I knew that chance, necessity, the combination of the two didn't work. I studied them carefully. Nobody disagreed with me, not even the good Professor Dawkins as late as last year. But there is a cause that we know can produce digital code. That cause is intelligence. We're doing it right now. I'm conveying information to you as maybe not an intelligent design, but at least some kind of mind is behind all this communication, okay? So what I realized, oddly, was that Darwin's own method of reasoning, where we infer the cause which is known to produce the effect in question is the best explanation of the thing we're trying to explain, that his method actually revived the argument from design. The data were different, the conclusion is different, but using his method, which I honored and followed, I came to the conclusion that the best explanation of the digital code that's involved in life, the central feature of living systems, has to come from an intelligence. And that is the basis, not of just a critique of evolutionary ideas about the origin of life, but a positive case for intelligent design based on an established scientific method of reasoning, in fact, Darwin's own method. That's my story.
Wonderful. All right.
I'll go to David.
Yeah. All right. I want you all to hang in there because what we're going to do in these next 17 minutes, David Berlinski, who is here with us, is going to basically do a little interview right now with Steve. And I want you to stick with this. We're going to do this for about 15 minutes. Then we're going to bring up Mike and maybe Ray and do a short Q&A. Are you guys ready? Come on out here, David. Come on out here, Steve. This is going to be a real treat. If you've, we're going to forego the video introduction of Dr. Berlinski, and the reason I want to do that is to maximize our time with him. Let me just recommend that all of you go and watch the movie Expelled. And it does a great job of introducing Dr. Berlinski. I'm grateful for his being here. He's come all the way from France. And so, Steve, I'm going to let you have a few minutes with him, then we're going to come back up here and crank out some more great time. So
that's great.
Welcome.